Hey guys, welcome back. The purpose of this video is to give you an introduction to the next lesson, 02.03 Early Cells. One of my most favorite topics, plant animal cells, prokaryotic cells, eukaryotic cells, and bacteria. So let's go ahead and get into the lesson so you can see what all you will learn here. So go ahead and click on 0203. Like always, scroll down to the bottom to find that Early Cells Biology Notebook. So again, you can click on it here and open it up if you'd like, or you can go over to our Google site, click on Module 2, scroll down, and find the Early Cells lesson. And here is the biology notebook for the Early Cells lesson. Okay, so you can see there's various things that we're going to be looking at. We've got a little bit of some background here on some scientists. We have something called the cell theory. Then we get into our types of cells, our prokaryotic and our eukaryotic cells. We're going to look at the cell membrane, and then we're going to wrap up this lesson by talking about selective permeability. Okay, and then again, you have a couple of sample questions here at the bottom. Okay, so let's go back to the top. Alrighty, hopefully I'm not making you sick with all my scrolling here or scrolling too quickly. Let's go back over here to the lesson. Okay, make note that we have 6 slides that we're going to look at here. Okay, there's a lot of interactive stuff in this lesson, so make sure you're taking time and care and looking all over the screen for all those things that you need to click on. All right, so our first portion of the lesson is called, Is It Made of Cells? And you have a little activity here where you're going to check mark whether you think something is made up of cells or not. Okay, and continue and read on to the little paragraph after that. Once you've finished this slide, continue on to slide 2. And now you're going to learn about the cell theory. Maybe you've heard of this, maybe you haven't. The cell theory has 3 key points. And we're going to go back and talk about this guy here again, this Anton von Leeuwenhoek. And we have a little timeline here on kind of the history of cells. So you can click on, we go all the way back to the 1600s. We've got 1655, 1833. We kind of have a big gap there. So you'll continue on through the timeline until you get all the way to the end. And the end is 1862. So all the while going through the timeline, make sure you're coming back over here and documenting some of that information, because there are notes that you will document on your note sheet here from that timeline. Here we've got Leeuwenhoek, we have Hooke, we have Brown, we have Schleiden, Schwann, Roelicher, Virchow, and Pasteur. I always have trouble with the von Roelicher name. Okay, so once you're done with this slide, then continue on to slide 3. Okay, in slide 3, now we're going to get into the comparing the different types of cells. Now, on this first page, there's lots of things to click on, so please take your time, read through things thoroughly, and document important information on your note slide. Okay, on the prokaryotic cells and eukaryotic cells, we have arrows here that we are going to view 5 different pages here and looking at some different cell parts. Okay, so make sure you read through that and look through each slide or each picture and clicking on that little purple arrow. Okay, once you're done with that, then we're going to look at just prokaryotes individually. And then we will look at eukaryotes, and then we have a cell types review. And then you've got a little activity here. It says select the 6 topics and drag them into the appropriate category. Okay, so you will do that and document this information over here on your notes page. So come back to the types of cells and document this information as you go through. All right, so next thing, we'll go on to the cell membrane, and we're going to revisit the topic of lipids that you had back in a previous lesson. Okay, so when we look at the cell membrane, we have 4 components here: the phospholipids, proteins, cholesterol, and carbohydrates. You can click on each of these and you get a little bit of information as to what their role is in the cell membrane. There's a little bit of information down below the picture on homeostasis. So once you've read through all that, and as you are reading through it, make sure you come back here and document that information on your notes page. Okay? And then we have another slide here on selective permeability. Here you are going to compare a couple different ways things can go in and out of a cell. Okay, we have two processes called endocytosis and exocytosis. We're going to look at passive and active transport. Okay, and you have again some tabs here. We've got a tab that describes passive transport to you, one that describes facilitated transport, and then one that describes active transport. Okay, and I'll just give you a heads up here. Facilitated diffusion is super important in the assignment that you're going to do on this lesson that you're going to turn in for a grade. You actually are going to do another lab report, and it's all about facilitated diffusion, because facilitated diffusion includes osmosis, the movement of water across the cell membrane. And that's what your lab report is going to be about, osmosis, and how water moves across that cell membrane. All right, so we will move on to slide 6 here. Now, like some of the other lessons on slide 6, you get a summary, and then you get the description to what the assignment actually is. So now this one, it is a lab, and you have a couple of options on this lab. You can view a video for the lab and document information from the video, or you can actually do the lab. So we have the video lab report, and whether you choose to watch the video or do the lab yourself, it's still going to be the same handout. Okay, so it says Cell Transport Hands-On Lab. We're going to need to come up with a title, an objective, a hypothesis, variables. Here's the materials that are listed. Here's the procedure, so you can read through that to see what it's going to be about. We have a data table. In our data table, we're going to look at time. So this lab, if you were to do it on your own in real life, which you can if you'd like to, you don't have to though, you can watch the video. It'll take 72 hours to do the lab, and technically take a little bit more because there's a little bit of an extension on this lab. You're going to document 2 observations for each time, and then document a circumference. Okay, and all of this information, if you choose to watch the video, it's all given to you. You are shown pictures, you're shown a ruler for you to document this data. Okay, and then we have some conclusion questions. We have 5 conclusion questions. Okay, so at this point, you would think that you are going to be done with the lab. Okay, but you're not actually done yet. There is a refining the experiment portion. Okay, so on the refining the experiment portion, let me come back to that in just one second, because let's talk about the lab a little bit more. Okay, here is the video that if you don't want to do the lab yourself, if you want to watch the video, you're going to watch it here. To give you an introduction what the video is about, it's what we're going to do is we're going to use eggs. We're going to pretend in the video, they're going to show you, they're going to take the eggs, they're going to put them in vinegar. Vinegar dissolves the cell membrane around the egg, okay? But it leaves that really thin, or I'm sorry, the vinegar dissolves the shell off of the egg, but it leaves that really thin membrane on the egg on the inside. Okay, I want you to think about when you hard boil an egg, and you go to peel off the shell, sometimes there's a thin membrane that stays around that egg. Okay, well, in this lab, that membrane is still there because the eggs we're working with are actually raw. So, very carefully, what we would do is we're going to submerge an egg into vinegar. We're going to let the vinegar dissolve the shell, and then we're left with this raw egg that has this thin membrane around it. It's kind of like a water balloon, but we want to be really careful because it has a raw egg in the inside of it. Okay, so what we're pretending then is that egg is a cell. That membrane that's surrounding the egg white and the egg yolk is like the cell membrane. Okay, we could pretend that the yolk is like the nucleus and that the white is like the cytoplasm on the inside. Okay, and then we're going to take that egg and we're going to submerge it in water, in distilled water. And we're going to see what happens to that egg over time. Is that egg going to take on water and get bigger? Or is that egg going to shrink and get smaller because it's losing water from the inside of the egg? So that's what you are to take a guess on. You're going to write a hypothesis as to what's going to happen to this egg the cell when we place it in distilled water. Okay, and as they go through the video, they're going to show you doing that process, and they're going to pretend to have taken measurements. Well, they don't pretend to take measurements. They actually take some measurements and they show you the ruler for you to record some dimensions. Okay, now, so once you have documented all of that, you've documented your hypothesis, the variables, the data, you've answered the conclusion questions. We think that we're done, okay? But actually we're not quite yet. There is a, let's see here, there is a refining the experiment portion of this lab. Okay, so if we scroll down to the bottom, the refining the experiment says, let's continue this experiment by removing the egg from the water and then putting it in syrup, like maple syrup. We're going to let it sit for 24 hours and then make some observations. Okay, so here's the hard part is they don't give you a video for this. The only way you can do this lab is if you search some videos on your own or if I give you the data. And so that's what I've done. I've also given you the data. Feel free to search some videos to see what might happen to an egg when you place it in maple syrup, pancake syrup. But you can also use the data that I've provided as well. Okay, so the first thing that you're going to want to do is take your best guess on what you think is going to happen to that egg when you place it in the syrup. Okay, and then you have a couple of questions here that you will address. How will the change in syrup within the egg be measured or identified? How will you determine when homeostasis has been reached in the egg and the syrup? And then you'll go back and your new prediction here on what you thought would happen to the egg and the syrup, you're going to evaluate that. Was your hypothesis right or not? And remember, it's not a bad thing if your hypothesis is incorrect. And how much did the egg change in size in centimeters? Okay, so for this refining the experiment, you have to use our Google site. So if we click over here to our Google site, remember there's our biology notebook. Here's the lab report that you can open up and make a copy. There's the grading rubric for you. Okay, if we scroll down just a little bit more, here's the data. Okay, so you'll find when you observe that video, here's the data of the egg and water. Okay, and I've provided you with data for the egg and syrup. All right, so you can see what's happening there. Hopefully you didn't look at that too much so it doesn't skew what you think your hypothesis would be. I also do have a video here done by the Sci guys, Science at Home, the naked egg and osmosis, that you can view if you need a little bit of extra help. Okay, so sorry for this video being too long, but I felt it needed a little bit more explanation because this one's a little bit more complicated than some of the other labs that you have done. So please, like always, send me messages, send me emails, so I can help you with anything here. Alrighty, guys, have a great day. Bye.